will be talking a little bit about art science, but particularly from my own perspective as an art science practitioner. And through sort of describing a lot of my works, I will be hoping to tease out some of the values that I believe, that are some of the key values of these interactions, but also the way that I think that my practice operates in terms of communication. And it's not strictly science communication, which is something that you'll see as I talk. But before I get on to talking about my own work, I'd just like to give audience members who might be a little bit unfamiliar with the idea of BioArt and art science a little bit of a background. So when I sort of talk about BioArt, I'm really talking about, and again, it's still an emerging term, but it's typically used to describe art practices that are using living organisms or using the manipulation of living organisms, and also using biotechnologies in their production. Art science is a more recent term again, and it sort of often describes projects that integrate the disciplines of art and science. And these projects are also often collaborative, and you'll also find that the boundaries between art and science and between the outcomes of those disciplines often become blurred as well. So while BioArt and art science are quite emerging in terms of an artistic genre, their lineage can still be traced back to the 1930s, where you have examples of people like Alexander Fleming, who did his microbial drawings. Alexander Fleming, of course, was the discoverer of penicillin. You also have one of the first sort of artworks that was exhibited that were genetically augmented delphiniums produced by the photographer and artist Ebert Steichen. And that was also in 1936. But it was only really in the 1980s and 90s that BioArt really became a more recognised term through pioneers such as Joe Davis and also Eduardo Kratz, and they produced a number of BioArts projects. And then, of course, later in 2000, you had the emergence of, again, new works such as, of course, the famous genetically modified bunny, the Alba, the GFP bunny, which again may or may not exist. And then you also have things emerging like MIA, the semi-living artist, which was a biorobotic installation, and the semi-living sculptures of the Tissue Culture and Art project from Perth. So just to give you a little bit more of an in-depth overview of some of these projects, and one that's quite closely related to my own practice, which is in cells and tissues. This is sort of a work that I really like by the Tissue Culture and Art project from 2002, which was called Pig Wings. And it was a project that was largely exploring the idea of xenotransplantation, which was very sort of popular at the time and really sensationalised in the media, which was kind of this idea that you would create transgenic pigs that had human DNA in them so that you could easily harvest the organs from those pigs and implant them into people that needed organs. And so what Oren and Yonat did was they actually grew these pig wings in the shape of bat, bird and pterosaurus wings. And they were sort of interrogating the sort of potential values of these technologies, and where they were using these kind of these kind of wings that had particular cultural associations. So you had the bat for bad and evil, the bird for, of course, angels and good, and then the pterosaurus, which were kind of still largely free from kind of cultural ideas. And they exhibit these sort of small tissue engineered sculptures in the laboratory, in a kind of art space, in a kind of laboratory type setting. And what I think was really nice is, of course, this work was talking a lot about the kind of idea of science making the impossible possible, so things like flying pigs, chimeras, things like that. But another thing that's really charming about their work is that they often talk about the aesthetics of disappointment. And so a lot of people, when they go to see the works, really thought that they were going to see flying pigs and pig wings. And then what they see are these little tiny grotesque lumps in these petri dishes that look absolutely nothing like what they had initially imagined. And so a lot of their work also sort of both interrogates the kind of ethical implications of technologies and the potentials that exist, but kind of also looks at the limitations of the technologies as well. So in that way, it does kind of have a function of engaging the public in a debate and a critical kind of scrutiny of these technologies, but also kind of communicating some of the limitations that exist. So as I mentioned, I wanted to talk about Oren and Yonat's work as an introduction because they're very well established practitioners, and as I said, very close to my own practice, which is working with cells and tissues. So just to give you a little bit of an idea, I'm a visual artist. I come from Australia, and I've been working within the tissue repair and regeneration group at the Institute of Health and Biomedical Innovation, sorry about that, for 5 years. And during this time, I learned a lot of the cell and tissue culture techniques. And here in the slide, you can actually see one of my first cells that I started working with, which are SAS2 cells, which are a bone cancer cell line originally derived from an 11-year-old girl back in 1973. And they're sort of cultured quite regularly in labs all over the world. Cells are grown mostly in a monolayer, and these cells are adherent, so they adhere to the bottom of the flask, and they're grown in the flask in a liquid nutrient medium, and then they're maintained in sterile conditions, also in incubators that simulate the same conditions inside of a body. And so when you're also working with the cells, they kind of require this ongoing maintenance, so that every 3 days or so you have to split the cells. So they grow to fill the available space in the flask, and then you need to split them into multiple flasks so that they can keep growing. And so what I became really interested in was just kind of looking after and caring for these cells. And what I really discovered is that, you know, it really challenged my perceptions of cells in terms of kind of, I really got to sort of see them as being really unique and independent organisms, but also that they're really connected to the environment in which they're living. So I was sort of, for the first 6 months, just culturing these cells in different environmental conditions and really observing the changes to those cells as well. And here, down the bottom here, you can actually see these are all SAS2 cells, but they're all looking quite different because of the different culturing conditions that I exposed them to. The other thing that I learned about cells is that the more that you culture them, so if you passage them for a number of times, the more you kind of have these mutations and things that start creeping into them. So that you kind of have these cells that kind of increasingly become more differentiated from the original cells that you were using. And that's why research scientists won't tend to use old cells or cells that have been passaged a number of times in their experiments, because then the experiments won't be reliably replicated. So why am I telling you all of this information? Because for me it was absolutely amazing, because I also kept thinking about the girl, this Alice, who existed back in 1973 and was most likely long dead because of the virulent nature of the cancer. And yet her cells were living on. And the total biomass of her cells would actually now far outweigh her body if she was actually still alive. And so for me, that was a really kind of uncanny sense that I kind of got from working with these cells of an absent body. The other thing was also that you have these cells that are continually kind of becoming other the more that you're passaging them, but at the same time they still contain the DNA of the girl, and so there's really kind of all of the information to recreate her body. And so for me, these were all really quite amazing things. And from that experience, these are some of my first works that I created, and it was part of a show that was eventually turned into about 7 different manifestations of kind of interconnected works. These are some of the first works, and it was part of a show called The Absence of Alice. And so the first work here was called Fragments of a Body in the Process of Becoming Other, which is the sort of work with the perspex, and on the wall, 78 impressions of a single object. So 78 impressions of a single object was really talking about the number of times that I had passaged Alice's cells. So I passaged them 78 times, and in that time really observed them changing. And so what I did was I actually made a sculpture of sort of like a bone tumour, and then I took 78 impressions from different angles. But also because the plaster was set and sometimes not a set, I got 78 completely unique impressions from that particular object. The more impressions that I made, the more of the plaster started building up on the object so that they become different again. So this work really sort of talked a lot about this kind of idea of continual change, but also the way in which you know, the impression that you get of a particular object or organism really depends on the particular methods that you're using, but also the particular temporal moment in which you take that snapshot. And the other work here, Fragments of a Body in the Process of Becoming Other, was then linking with that work again, where I kind of took latex impressions of the impressions on the wall and mixed them with the actual cell palettes that I'd collected during the passages, and then displayed them on the slides. And the intention with this was again to kind of talk about this idea that science likes to fix these moments in time, even though you have an organism that isn't a kind of continual state of change. But then I also wanted to kind of create a linkage to this kind of body, but a body that was sort of no longer a body the way that we know it. So it had these kind of a real fleshy kind of sensibility as well when you looked at it. So those were some of my first works. Later on I went on to work with other cell lines as well, and this is a work called HEC293T, The Transformation of Joni or Oliver. And this was a work that I developed and researched and developed in collaboration with researchers and staff at Symbolic, which is a dedicated art science laboratory at the University of Western Australia. And this work, I was working with human embryonic kidney cells, which is what HEC stands for. And human embryonic kidney cells were really interesting to me because they're actually a cell line that was established from an aborted fetus, and it was actually modified with the adenovirus DNA, so shared adenovirus 5 DNA, so viral DNA, to make it that it was immortal, so that they would keep dividing indefinitely. Most cells, if I isolate cells from you directly, will only replicate about 50 times until they reach the Hayflick limit, and then they'll stop. Whereas immortalized cells will essentially are like cancerous cells, so they'll just keep dividing indefinitely. The interesting thing about the HEC293T strain is that it's been further modified with the large T antigen of the simian virus 40, which means that it also makes the particular cells more easy for genetic augmentation through the addition of additional DNA from different organisms. So at Symbolic, I worked with the HEC293T cells, and with the help of Jill Mewling, who was one of the molecular biologists there, I was able to also further augment those cells by introducing red and green fluorescent proteins from marine invertebrates. And so for me it was kind of interesting about the kind of origins of the cells, but also the way that we're really, through biotechnologies, really complicating the kind of taxonomic categories that we have traditionally established. But that you also kind of have both horror and wonder that are always really closely entwined with these technologies. So the work that I created was kind of like this tree that was growing out of bone, and it had teeth and blood and flesh and hair and flowers that looked like they were skin, and these little kind of fetuses that were pods that butterflies were coming out of. And so it was kind of both magical and really disturbing at the same time, because I kind of have that sensibility in my engagements with these technologies. I also had the original cell class, which you can sort of see in the central image there, with a video underneath that kind of talked a lot about the origins of the cells. Also had video footage of the actual glowing cells when they were transformed, etc. So that was sort of another work, and again kind of looking at the uncanniness of the technologies and aiming to also impart some information about what was really happening in the lab as well, and ask questions for the viewers. That work also led me into the development of sort of another work here, which was called the Immortalization of Kiran Rama. And that was where I became interested in the actual products that are used in cell and tissue culture as well. So when you're actually growing cells in the flask, in the liquid nutrient medium, often you also have, you always have the inclusion of fetal bovine serum, which is a protein-rich nutrient supplement which is added to the medium in order to keep the cells alive and viable. And so I became really interested in where they actually get this fetal calf serum from, because it's actually derived from the blood of fetal calves. Where do these fetal calves come from? And I did have a moment where I did imagine that there'd be like Matrix-style vats filled with cows that are having their blood routinely sucked out. And that was a really disturbing image. So I actually investigated where these calves came from and how they harvested the blood. And that's where I discovered that they're actually often just a byproduct from meat production. So that's why calf blood is used quite regularly and used to make the fetal calf serum, because pregnant cows are slaughtered the same as any other cow, only here they'll drain the blood of the fetus, but the actual body of the calf is discarded because it's deemed unfit for human consumption. So for me, I was kind of interested in these kind of unseen victims of Western consumption that are both within labs, but also within our everyday consumption of meat and other sort of products that we don't really think about and we don't know about. So the idea of this project was that I would create a bioreactor that would, first I'd isolate cells from a fetal calf, immortalize them with viral DNA so that they could essentially live forever in the right conditions, mummify their bodies, and then display the mummified calf fetus with the living cells, and then also feed the mothers of the calves to people as they looked at the work. So that essentially it would kind of interlink these kind of the idea of consumption and the unseen victims of consumption, but then also look at the way in which new technologies are really changing our perceptions of life and death, but also really sort of, so shifting all of these boundaries. So I did end up going to Symbotica, and this was another project that I did in the same residency. And I got 2 fetal calves, Kira and Rama this time, and during the residency I was actually able to isolate cells from the calves. And that's just some images of getting the calves and isolating them. They were really big as well, and it was quite a traumatizing experience. But from that experience, I did end up making a bioreactor, and this was the bioreactor prototype, which was shown as part of a larger exhibition that looked at human and animal engagements at the block in QT. And it kind of consisted of 3 components. So there was the bioreactor component, which had like a video that sort of talked about the process of isolation and the history of the calves. And inside you can sort of see here, there was the sort of bioreactor, which is again a prototype, but it worked. And what I did was I actually reanimated Kira, so the female calf cells, and seeded them into a silkworm cocoon, because that way it could actually have a 3-dimensional structure, so they were living in the cocoon, and the bioreactor was basically just keeping the cells alive in a kind of artificial body. I also had a lot of these sort of symbols of kind of transitioning between life and death. So here you can see a raven, and the raven was included as kind of a symbol of like, because in mythology a lot of ravens are used to actually helping the souls transition from death to life and backwards and forwards. But I also had other things included, so relics from sort of from the cell isolation process, so the initial cell culture vessel, trials from when I was seeding the cells into different scaffolds, and also the cryovials that I had isolated from Kira as well. There was also engraved objects with poetry as well, but I won't have time to go through that. In addition to the bioreactor, I had, instead of the mummified bodies, which is a very funny story, you can talk to me about that later, of why they don't exist, but I actually then had these relics from the isolation process, which were also shown in conjunction. So that was things like their hearts, their teeth, slides of their various organs, things like that. And that was included to create a really tangible and visible link with the actual bodies of the calves. But I also kind of created these small dioramas, which you can see here, which kind of came from a previous exhibition and then carried over. And they were sort of made of these miniature landscapes, which kind of were composed of the tanned and salted hide of the calves, with little miniature chairs representative of the 2 calves that also had wings. And so these were kind of talking a little bit more about this kind of magic in a sense as well, and kind of the transformation of the calves. So the wings had like butterfly wings, but also little skulls painted on them, so that it was kind of representative of transformation through death. But I had these kind of magical elements included there as well, so there was kind of the real viscera and the kind of horrific elements, because for me the process of cell isolation was both magical and really horrific at the same time, and I kind of wanted to give a little bit more of that sense to viewers as well. And I did make pies of the mother's meat and fed them to the people at the exhibition opening, and they did get eaten, so everyone ate them quite proudly, I think. These works were also shown in conjunction with other works that, as I mentioned, talked about the relationship between humans and animals, and was particularly kind of looking at the way in which cultural beliefs and those sorts of things really influence our perceptions of value in terms of which organisms we are valuing, which organisms we don't. And so this is one work that I think really quite sums up sort of the types of other works that were included. And it was called The Influence of Aesthetics on Ethics Demonstrated in a Spider Eating a Butterfly. And it included a large sort of like sculptural installation that included a terrarium in which there were living spiders and living chrysalises, so that when the butterflies would hatch, the spider would come and eat them. And that also included a poem, which I won't read out to you, but the poem was really kind of talking a lot about how we really approach everything from very much an anthropocentric viewpoint, where, and also where we kind of privilege some organisms over others, but really, especially with the emergence of new technologies and things like that, we really need to also start to appreciate all organisms, whether they are typical organisms, mutant organisms, genetically engineered, so-called natural organisms, but they all need to be approached with a sense of care, I guess. Finally, I'll just touch on 2 more projects and then I promise I'll be finished because I think I'm running out of time. But this is one of my more recent projects, and this falls a little bit more into science kind of engagement, science communication in a sense. But this was a project where I was really working with my group at a university that works with wound healing, and so they actually develop a number of therapeutic models to look at improving health and well-being, particularly of burns victims, scarring patients, etc. And as part of their research to reduce the number of animal models, they've developed human skin equivalent models, which are made up of the surgical offcuts of consenting patients. And so for my project, I decided to use the same technology, but actually make it jewellery out of it. And that was kind of deliberately provocative when you say I'm making skin jewellery, because of course it immediately gets people going, oh, I don't really agree with that. A lot of things, you know, sort of come bubbling to the surface in terms of people's taboos surrounding the use of cells and tissues and the use of body products for ornamentation as well, particularly. So this project was really kind of looking at also making these kind of provocative objects to get people kind of interested, but also to allow participants to have a first-hand engagement with the technologies that they might never actually previously have the ability to interact with. So for my project, I actually ended up donating skin from my arms, which was used as a dermal scaffold, and then collecting hair from the participants, which were then had skin cells isolated, seeded back onto my skin, and they were grown up as the human skin equivalents, and then later they were fixed and stained. Over here you can actually see one of the human skin equivalents with the cells on it, and it's stained with MTT stain, showing the cellular proliferation. So once they were fixed, we fixed them in formaldehyde further, and then they got set in resin. And I worked with the participants to create a jewellery item that then kind of captured their experience and their engagement of that project. And that was up to them as to what they wanted their jewellery piece to represent. And so this project was also about kind of allowing the creation of an artwork that would exist outside of a laboratory setting, so that you could actually wear it in an everyday encounter and talk about your experiences with your friends, and also have a real personal connection to the project as well. But the other thing that I was interested with this project is having some outcomes for the scientists as well. So during this project, I was able to establish a standard operating protocol for isolating hair follicle keratinocytes, which hadn't really been done at my lab. It wasn't a new protocol because it has been established previously, but I've now got it running very smoothly at my lab, and I've had most of the scientists in my lab have now had their own cells isolated, and they have images of them, which is kind of really nice to get them really invested in the technology. And the other thing was testing the HSE growth in serum-free conditions, so without animal products, because one of my participants was vegan. So she used human products instead of animal products, on the grounds that, that humans can make the choice, which again, I had to say to her, sometimes in biotechnology not so much, especially with the history of cell and tissue, it sparked these interesting debates as well. So the next project that I'm working on takes this engagement across art and science a little bit further, and this is where I'm working on the Biosolar cells project here in Leiden. And that's a project that's looking at complementing natural photosynthetic processes for potential future energy production and the production of biofuels. And with this project, what I'm really interested in is trying to be able to contribute to the actual research in the lab while also gaining skills and then later making artwork. So I'm really interested in trying to create a space where you're not just using the resources of the scientists, but in some way contributing to their outcomes. Because I think a lot of the time I felt like I was a bit of a drain on resources when I was in my early stages of learning about working in the lab and culturing cells. So here I'm really interested in creating projects that might have multiple outcomes. And that concludes, I think, most of the things that I wanted to talk about. But I just wanted to, at the very last, sort of summarise some of the key things that I think are important in terms of art and science, intersections and collaborations. And one of the things that I think is really important to keep in mind with all of these things is that I've sort of talked a lot about my project, but I think that there are so many different types of projects that exist within the kind of realm of bioart and art science that we have to kind of always accept that they have multiple outcomes, multiple potentials, and also multiple resonances. So for me, working across art and science has provided new tools, subjects, and also methodologies, which have really both transformed my practice, and I think has also had a transformative kind of operation within a lot of the scientists that I've been working with, in terms of thinking more about the origins of their cells and the materials that they're using, and also starting to think more about the kind of ethical implications that extend beyond their scientific work. I think it also allows non-scientists to learn and also engage with contemporary technologies. It challenges a lot of disciplinary assumptions, and it kind of facilitates other ways of producing knowledge, particularly within science. It does function in part, you can see, as science communication. So in all of my works, I do talk about the science, and I try to be as accurate as I possibly can in terms of what I'm presenting, but it's not PR in my case, absolutely not. So it functions more as a dissemination of contemporary scientific practices, but it always has this kind of exploration and critical engagement as well. Other things are things like connecting it to personal and emotional attentions, drawing attention to the way in which scientific practices and beliefs are also connected to culture, cultural attitudes, societal norms. So basically, it's a critique also of kind of this idea of objective knowledge. I also think that in a lot of cases it also creates an imaginative and a playful space to think about things that might not exist yet, so that we can think about possible futures without being restricted to what already actually is there. So it opens us also to these new expectations of possible worlds, and I think that scientists and artists would agree, I think both, that we really can't anticipate the way that our technologies will play out in the big scale in terms of the future. But I think it's really good to start thinking about all of these possibilities, even if they don't come to be, because we need to be prepared for the unexpected and unexpected outcomes. And finally, I think also art-science collaborations do have the potential to have scientific merit and contribute to scientific research objectives, and therefore can contribute potentially to outcomes across both disciplines in the arts, in the sciences, and other disciplines as well. And I'd just like to, again, say a quick thank you to all the people that have supported me. So as an artist working across art of science, I have a number of different institutions and people that are fundamental in helping me realise a lot of the ideas and providing guidance and assistance. So thank you to QUT and IBI, my supervisors, visual arts technical staff, Symbolic, my PhD project participants, and also the Art and Genomics Centre, Leiden University, and the Biosolar Cells projects, for allowing me to come here to Leiden and to continue my work. So thank you.